So our topic is physical mapping of genes and DNA sequencing. First of all, DNA mapping is a process by which we determine the genes and their position along a given chromosome. The distance between two genes is typically given in map units or recombination units. To calculate the map units between two genes, we must determine the percent recombination between two genes.
Okay, next one is DNA sequencing. Is the process of determining the sequence of nucleotide bases in a piece of DNA. Today, with the right equipment and materials, sequencing a short piece of DNA is relatively straightforward.
So regions of DNA up to about 9 million base pairs in length are routinely sequenced using a method called Sanger sequencing or the chain termination method. Sanger sequencing was developed by the British biochemist Fred Sanger and his colleagues in 1977. So, first of all, we need
some kind of ingredients, such as a DNA polymerase enzyme. The second one, a primer, which is a short piece of single-stranded DNA that binds to the template DNA and acts as a starter for the polymerase.
So Sanger sequencing involves making many copies of target DNA region. Its ingredients are similar to those needed for DNA replication in an organ or for polymerase chain reaction that we learned in last term. Then next, this process. The DNA sample to be sequenced is combined in a tube with primer, DNA polymerase, and DNA nucleotides. The four dye-labeled chain-terminating deoxynucleotides are added as well, but in much smaller amounts than the ordinary nucleotides. Then the mixture heated to denature the template DNA, separate the strands, then cooled so that the primer can bind to the single-stranded template. Once the primer has bound, the temperature is raised again, allowing DNA polymerase to synthesize new DNA starting from the primer. DNA polymerase will continue adding nucleotides to the chain until it happens to add a deoxynucleotide instead of a normal one. At that point, no further nucleotides can be added, so the strand will end with deoxynucleotide. We can see it from these pictures, like here primer, template, and after adding those nucleotides, we can, those colorful nucleotides are binded to 3 ending, and we will see after can cut it and repeat this process again and again in order to write our genome.